So now in this part we'll start talking about the specification of limb types by two genes, the TBX4 and the TBX5. TBX4 and 5 are transcriptional factors that are expressed differently in the forelimb bud and in the hind limb bud. As you can see here in the chicken embryo, for example, in the in situ hybridization, to show the expression of the TBX4 and 5, we can see that TBX5 in blue is expressed in the anterior lateral plate mesoderm that will form the forebud, and this forelimb bud will be developed into wing, while the TBX4 in red, this gene is expressed in the posterior lateral plate mesoderm. So this TBX in this limb TBX expressed here will give a hind limb bud that will be developed into a leg. So in one of the experiments done, they injected beads, containing vectors expressing FGF gene. And they inserted the beads, right in this position between the regions of the TBX5 and the region of the TBX4. So the bead will induce the expression of TBX, as you can see. The beads will induce the expression of TBX4 posteriorly and TBX5 anteriorly, so that we can get a kind of chimera kind of limb, having TBX4 and TBX5, while the limb bud expressing TBX5 will induce the expression of a wing, while TBX4 limb buds will be developed into legs. So it seems like we don't have exactly the same gene expression in the forelimb bud and in the hind limb bud to induce the formation of wings and legs in chicken. So TBX4 is expressed in the forelimb bud, while TBX5 is expressed in the hind limb bud. And here I'm showing you a chicken embryo, and this is the chimera kind of limbs induced by the insertion of the FGF beads, so that we can have an anterior wing and a posterior leg, since we have this kind of TBX4 and TBX5 anteriorly and TBX4 posteriorly. So the gene encoding the TBX5 is transcribed in the anterior lateral plate mesoderm and in the forelimbs, as I explained before, and the genes encoding the transcription factor TBX4 are expressed in the posterior lateral plate mesoderm and in the hind limbs. So in order to know what makes the difference between the forelimbs and hind limbs in terms of gene expression, they tried to do many experiments. In one of these experiments done in mice, for example, or in chicken, they make a knockout, it means mutation, for either TBX5 or TBX4. In this case, the lateral plate mesoderm fails to make FGF10. You know that FGF10 is usually expressed in the lateral plate mesoderm. So in this case, FGF10 is not expressed in the forelimb or in the hind limb regions. So what does it mean? It means that TBX4 and 5 act upstream of FGF10. So they can induce the expression of FGF10. And more studies have shown that we have HOX, you see, HOX, the homeotic gene. HOX proteins can activate TBX genes, and these TBX genes will activate FGF10. So in a cascade, so we have cascades of gene activation in hind limbs and forelimbs, so that HOX can activate TBX and TBX can activate FGF10. So Interestingly, they try to do this experiment when they try to replace TBX4, okay? So TBX4, which is usually expressed, TBX4 in red, in the hind limb bud, and TBX5 in the forelimb bud. So they found that TBX4 can be replaced by TBX5, but surprisingly without changing the limb from hind limb to forelimb. What does it mean? It means that TBX4 is not the only gene that will define and specify the hind limb, and TBX5, it's not the only gene responsible for the specification of the forelimb, okay? Because if we replace one by another one, we cannot change the identity of the hind limb by a forelimb. Other experiments done in mice this time. So in mice, they found that we have one gene. It's the PITX1. This PITX1 is critical in the formation of the hind limbs. It's highly specific for the formation of the hind limbs. So they tried to overexpress PITX1 gene in the mouse forelimb, okay, and this results in the transformation of the forelimb into hind limb-like morphology. Okay, in mice, it seems it's different than it was the case in a chicken, for example, since when we overexpress PITX1 in the forelimbs, we can this is enough to induce the transformation of the forelimb into hind limb, since PITX1 is critical in the formation of the hind limbs. So this is what's known about the specification and the genes expressed in the forelimbs and the hind limbs. And it seems like TBX5 and TBX4 are not the only genes that specify the forelimbs and the hind limbs. So now I will talk about different models for the specification of the proximal distal axis. Why models? Because nothing is completely confirmed and nothing is completely sure about how exactly the proximal distal axis of limbs is established. So we have 3 different models for the specification of the proximal distal axis. In the first model, it's called progress zone model. In the progress zone model, you remember that here we have the AER, okay, which is the distal part of the ectoderm in a limb bud, and just under the AER you see this pink zone. It's the progress zone. It's a zone of high mitotic activity. So in this zone usually cells will proliferate. The red spot is a cell under proliferation in this progress zone. So according to this progress zone model postulates that each mesodermal cell is specified by the amount of time it spends dividing in this progress zone. So the longer a cell spends time in this progress zone, okay, the more mitosis it will achieve, and the more distal structures it will give. Okay, so it means, for example, here if we have cells 1, cells 2, and cell 3, and cell 4, the cell that will exit the progress zone first, it will give the most proximal structures, and then comes the other cells that will give the more distal cells. And here, for example, the cells number 4 that will exit the progress zone later, okay, where they will give more distal structures. Okay, that's why it's called progress zone model. The second model is called early allocation and progenitor expansion. As its name indicates, this model for specification of proximal distal axis of limbs, okay, stands it's based on the fact that the different territories or different zones of the limbs are already present very early, even at the level of the limb bud. Okay, you see we have the zone of the cells number 1, the zone 2, the zone 3, 4. And what happens during the specification and the growth of the limbs according to the proximal distal axis is that we have expansion. Okay, just expansion of these zones. We have enlargement and expansion of these progenitors, of these zones, in order to form the different territories of the limb. Okay, so it means that the territories of the limb bud are established very early, and the cells will grow in each of these areas formed very early after their expansion with time. Okay, so we have progress zone model, early allocation, and progenitor expansion. But you should know that these two models still have to show or to demonstrate how the progenitor cells of each region are generated. It's not known, it's not explained at the molecular level how these progenitors are specified into a specific or particular segment of the limb. So it means that we still need to know and to study how or to study the molecular mechanisms that specify the proximal distal limb regions. It means to know the genes that will specify the progenitors and that will later specify a particular segment of the limb.